We're ready.
Right. Hi, everybody. Welcome. Thank you so much for joining us for our fourth CLARiTI webinar. We've got a packed agenda, so let's go ahead and get started right away. Phoebe, if you could go to the next slide, please. All right, so first you'll hear from Drs. Johnson and Mormino and myself, Heather O'Connell from NAC team, just to go over some study updates. And then we have got the team from NCRAD and then an overview from Neuropath. Next slide, please. All right, so just getting a little bit into more detail here, we're going to talk about some of our study goals. Beth is going to go over our year one progress report that's due July 1st, covering the P30 memo that was sent out recently. Sterling's got some updates on the MRI protocol. I'm just going to cover a couple of site readiness items, and then we're going to hear from Heather discussing subawards, everyone's favorite topic. Next slide, please. All right, I'm going to hand it off to Dr. Sterling Johnson, our fearless leader. Thanks.
Thank you, Erin. Really appreciate you all joining today. Just wanted to quickly remind us all why we're doing this. First and foremost, CLARiTI is about giving you resources. It's an infrastructure grant, but our opportunity here, our scientific opportunity, is to understand the mixed etiologies that may be present in our participants, to try and detect that in living people. We'll confirm that with Neuropath at the end of life if we have that opportunity. And we'll talk about that today, but just wanted to bring us to center us on this important scientific goal so that we can better address the needs of our patients and our participants. And our goal over here in terms of the actual recruiting goal is 200 participants from across the ADRCs with at least 25% diverse representation from people who are from historically underrepresented groups. We hope to be able to detect these co-pathologies that are listed there in the lower left. Just wanted to bring that up as an overall perspective on why we're doing what we're doing. Let's go on to the next slide. And I'll turn it over to Dr. Beth Mormino.
Hi, good morning. So believe it or not, we're submitting already our first year progress report next week. So just to highlight some major accomplishments for this first year. So IRB and FDA approvals are in place. The performance site initiations are beginning now. And excitingly, we expect the first enrollee to happen next month at Wisconsin. In addition to study startup procedures, we've also coordinated across NAC, LONI, SCAN, and NCRAD in anticipation of this exciting incoming data that we're going to be seeing. And I do want to, in particular, acknowledge NAC, who has been developing additional workflows, to integrate what we're going to be doing in CLARiTI. So we also have a number of cores, as you know, a part of CLARiTI, and progress has been made across many of these cores in year one. So I'm just going to bring up a few highlights. So we've collected survey data related to site activities and needs related to inclusion and disclosure practices. So most of you guys helped us with those surveys. We also, within the visual interpretation team, they've selected 10 visual raters that will assess CLARiTI PET scans and provide centralized reads back to sites. And then we've also initiated a number of data leveraging projects using already collected ADRC imaging data as well. And then finally, we do have a large amount of our budget that was not spent, given that we haven't actually collected new data, but we'll be able to use that in subsequent years. Next slide, please. Yeah, and then hopefully you guys have seen this, but we did send around a memo. So this memo describes our plan to offer a CLARiTI infrastructure to support non-CLARiTI scanning that is going to be happening across ADRCs. So for instance, there's this new P30 requirement of 24 ATN sets per year that are separate from CLARiTI. However, we've worked out ways in which the protocol and the contracts with the ligand companies can be applied to those scans. So if you have more questions about that, feel free to reach out, and you can also read the memo for more details. All right, next slide. Okay, and then I'm going to turn it back over to Sterling.
Thanks, Beth. Just a quick update on the MR protocol. Some of you are asking about this, and especially as we get close to enrolling, we'll be solidifying the plan here. But just so you're all aware and that we're on the same page, if the scan is successful and considered complete, if you've done the scan T1 weighted volume and the scan flare, and those will be labeled as the CLARiTI T1 volume and the CLARiTI flare, and they'll be uploaded to a CLARiTI portal at Lonnie, but it'll be a very similar process and a very similar workflow to the regular scan sequences, but we'll be using those same MR sequences, those first two. Those are our anchoring common sequences that we do across the centers program that we all have agreed to do. But you'll have lots of time left during that hour, and we have given you options for that. You can do this, you can fill the rest of that hour with the remaining scan sequences, and we would encourage you to think about doing that. Those are really excellent sequences. We also know that you have locally prioritized sequences and continuity that you want to maintain, so you have flexibility there. And we also have CLARiTI advanced sequences that are emphasizing neurodegeneration, and that'll include an SWI QSM multi-echo gradient echo acquisition, and then this motion corrected T1 weighted volume to look at the impact of motion on the quantitative outputs that we get. And then there'll be some ASL sequences that we would like to run as well. So that's your third option, is to do the CLARiTI advanced sequences. And we'll have more guidance on this coming forward in the one of our upcoming webinars, and also in some written guidance in the near future. All right, let's go on to the next one.
All right, so getting back to site readiness. So again, I know I've shared this screenshot before. This is something that the NAC team is creating. We're hoping to have this snapshot available for all study sites. As you work through your site readiness checklist, working with your project coordinator, Kelsey or Hunja, make sure you're just keeping them updated on your progress with study startup. They're going to be tracking it at our end, and we're going to be releasing this dashboard next month when we have our next CLARiTI Connect newsletter come out. So that way you'll be able to see where your progress is and that of any other site as well. So hopefully that creates a little bit of a friendly competition. Next slide, please. And then we're also going to be releasing our CLARiTI study handbook as well. So, as you can imagine, as we're working through the study startup procedures, we still have a lot of updates, but we do plan to release our first version of the handbook again when we release the CLARiTI Connect newsletter next month. So that'll be that's scheduled for July 10th. Next slide, please. And then I just wanted to remind everybody, we do have new consent templates. So we have had a couple of sites that have sent in their consent documents, their site-specific consent documents for review, and they were our old consent templates. So just wanted to make sure everybody was aware. They're listed on our CLARiTI website under toolkits and resources. You can see the link there, IRB approved documents. That'll take you to a box folder. So you can see that we're on version 2 for both cognitively unimpaired and cognitively impaired. And then we have version 1 of our results disclosure consent. So we did take that results disclosure consent out of the cognitively unimpaired and made it its own based on some feedback from sites. So just wanted to point that out, making sure that you are submitting the appropriate consent documents. All right, next slide, please. And then we just have a couple of new study personnel. So Brittany Sloan, Senior Regulatory Coordinator. She's working on the admin core. Terry Ward, Neuroimaging Analyst, again at the admin core. And then Carol Sapp, she's a new Clinical Research Supervisor who will be working with Dr. Gil Rabinovich's team working on visual reads. And we'll have some bios in our next CLARiTI Connect newsletter as well. So you can learn more about our new team members. And now I'm going to pass it off to my partner, Heather from NAC. Take it away.
Hi, everyone. Heather O'Connell from NAC, the CLARiTI Program Manager. And I wanted to provide an update on subawards today. In February's webinar, I went through the overview on subawards and the full payment process today, just going to provide an update on the timeline. So pleased to share that we have initiated the process with Stanford as the pilot site for our process to work out the kinks. And the remaining sites that are participating in year one will be initiated in the coming weeks. So given that we're starting at this time, we are looking at the timing for execution in that September timeframe. So what it will look like when you hear from us, you'll get an email from us requesting documentation. Included in that will be the scope of work and budget and payment terms. And we will be requesting documents that will be submitted via a REDCap survey. So we'll be asking for your site F&A agreement, budget justification, institutional letter of intent, and your seed IRB approval letter. Next slide, please. A little more detail on the budgets themselves. On the right, I have an excerpt from the budget and payment terms document that will be attached to your email that you receive from us. This is a snapshot of what it looks like. The first sections of startup and personnel costs are standardized across all sites. Procedure costs down in the blue there are variable by site, and they were developed using the calculation of the site procedural costs that you provided on your startup survey and multiplied by that IDC rate provided on the startup survey to reach a cost. So that final cost is what you'll see listed there. And a reminder that, you know, when we get to the point of invoicing after data submission, we need to receive both the image and plasma as well as the CRF data. Those components are required to be eligible for payments for each of those procedures. So that is the update I had for you, short and sweet. I think we are ready to transition to the next speaker. If there are any questions related to these study updates that we provided, we ask that you drop them in the Q&A section at the bottom of your Zoom chat. But with that, I will hand it off to our presenter, Dr. Tatiana Foroud, who will be covering blood biomarkers.
Thanks so much, Heather. Really glad to be with all of you today. And my fellow partner in crime is Jeff Dage. So we'll be doing this together. I'll start and then he will take over for me. So let's go to the next slide. Okay, so here's what we're going to be covering today. We're going to cover the biospecimen submission workflow. We're going to talk about how to get CLARiTI collection kits, also the collection and processing procedures. Probably a topic that we'll spend a little bit of time on is how the CLARiTI protocol overlaps with the ADCFB, Alzheimer's Disease Fluid Biomarker Initiative. And then Jeff will take over when we talk about the blood-based biomarkers at NCRAD and the Biomarker Assay Laboratory, and also novel biomarkers and remote collection devices. So let's go to the next slide. Okay, so let me start with an overview. So this is a really lovely slide that was made by NAC and kind of goes through the different parts of this process. So starting with the biosample collection, for the ADRCs, you're going to order your kits. NCRAD, that's us, will be shipping you the kits. And of course, the ADRCs will then be collecting the biosample. When you think then, once you've collected the samples, what's going to occur next? Okay, you're going to be shipping the samples to us. That's sort of the column on the left. Then we'll be processing the sample. That's sort of the sample flow. To the right, there's of course data that has to be collected about how you collected that sample. So you're going to be collecting information in a CRF, and that will then get incorporated and integrated with the other data at NAC. So that's all the site-related part. And then when we think about what's going on in terms of data sharing, how will results get back to the ADRCs? So on the left, you'll be able to access results through the NAC ADRC portal. There'll be specifically CLARiTI dashboards that will also be tied then to that payment tracking dashboard. And then for other researchers, so other researchers who want to be able to use data generated from the plasma samples, they'll be able to access those data from the NAC data front door that's being developed. There'll be a quick access file, and there'll also be a way to do cohort selection. So there's a lot of work, and part of what you may have questions about is how will this integrate again with the ADCFB, and we're going to cover that in a little bit after we go through some kind of basics about sample collection. Let's go to the next slide. Okay, so what here, there's hopefully is no surprise here. Again, what we're collecting is blood that's going to be for a source of DNA and for a source of plasma. You'll order kits on demand. You'll want to have a supply of kits available at your site. The link is right there, and these slides will be distributed. Showing you on the right is exactly what this is going to look like. So CLARiTI kits contain the collection tubes that you'll use, the cryovalves that you'll use for aliquoting, the labels that we'll want you to use, and all the materials that you're going to need for shipping, which you can see on the right, except for dry ice. Importantly, please allow up to 3 weeks when you ask for kits for those to be received. So there's no reason, you want to be able to have a little bit of a stock there. Make sure that that's always filled. We'd hate to have you not have kits available when you're ready to see a participant. Next slide. So this is the protocol. It's small. I'm going to give you kind of a high-level overview. If there are questions, we can certainly get into that. What we try with the schematic is to make it as visual as we can, so that for those of you visual learners, but we also have obviously the details in the text below. So starting from the left, you're going to store those tubes at room temperature. We'll often get questions about that. Each tube is going to be labeled. We've got collection tube, we've got site and PTID labels. You're going to have all that on there. Important that you use our labels. I'd say don't use labels that you've got at your site. Please use our labels. You're going to fill these tubes, allowing the blood to flow for 10 seconds and ensuring that the blood flow is stopped. You're going to immediately after the blood draw, you're going to invert those tubes and turn them to mix properly 8 to 10 times. You're going to want, after it's thoroughly mixed, you're going to want to put those samples on wet ice until you're ready to do centrifugation. We realize that depending on where you're doing this collection, sometimes you need to move those samples to where you have access to centrifuges. You're going to centrifuge those samples at 200 G for 10 minutes at 4 degrees C. Then you're going to start doing your aliquoting. Again, you're going to be pooling your plasma from the 3 EDTA tubes. Remember, you're doing 3 collections there. You're going to be inverting that again, make sure we've got everything mixed, and then you're going to be ready to do sub-aliquoting. Again, you've got labels. We've got caps that make it easy for you to see what goes into which labels. We've got a clear capped cryoval. That's where you're going to put the Buffy coat. You're going to get a Buffy coat out of each of those 3 EDTAs. And on the top, you can see that you've got your collections to go with plasma. Notice those all have purple caps, except for one right there. That's a blue cap. That's going to be your residual sample. So we realize you may not be able to fit properly. If you have a non-full collection as that last one, that's what you're going to put in there. And you're going to have 1.5 mils in each of those purple top tubes, except for that last residual. Hoping that that's all clear. You're going to store those plasma aliquots at -80 until you ship them to us. And hopefully I haven't gone through that too quickly, but if you've got questions, glad to take that at the end. Let's go to the next slide. Okay, so for some of you, you're already participating in what we call the Alzheimer's Disease Fluid Biomarker Initiative. I'm flagging on the left all the sites that are participating, and what you can see, a core part of ADCFB is that we're getting a source of DNA, and we're also obtaining plasma. So what you can see is across the sites that are participating, we've received over 9,000 subjects and visits with plasma. So that leads us, of course, into how do these initiatives coordinate? And let's go to the next slide. So hopefully this schematic will make it clear how we can coordinate, but also the importance of doing each of these initiatives. So on the left, we've got ADCFB. On the right, we've got CLARiTI. And in the middle, we've tried to explain what's in common across these initiatives. So for the ADCFB, it's a total blood volume. It does vary by site from 10 to 40 mils of blood being collected. For CLARiTI, it's a 30 mil blood draw, 3 purple top EDTA tubes, as I showed you on that earlier slide. For ADCFB, some sites are also providing PBMCs and CSF. That is not what we're doing in CLARiTI. CLARiTI is a plasma and Buffy coat for DNA. That's what we're collecting. Importantly now, we'll kind of walk through, again, some of the differences there. ADCFB, sites and participants are not receiving a payment. As you recall, that's a newer development. For CLARiTI, sites and participants may receive payments from NAC. For ADCFB, plasma biomarkers are paid by NCRAD. For CLARiTI, the plasma biomarkers are paid by CLARiTI. So let me walk through sort of what that has in common. So both those initiatives are collecting plasma and buffy coat. Both initiatives are going to obtain ApoE from the DNA, and also GWAS is going to be obtained from that buffy coat, the source of DNA. Both initiatives are going to get plasma biomarkers generated in the same laboratory in the NCRAD biomarker assay laboratory. And you're going to be able to access that data, both the ApoE, the GWAS, and the biomarker data through the NAC data portals. We're going to have CLARiTI specific portals, and we also have ADCFB, but importantly, they're all going to be in the same place. All that data is going to be generated in the same way, so it'll be able to be seamlessly integrated. I want to stress the ideal solution is for individuals to participate in ADCFB and for individuals to participate in CLARiTI. The reason for that, just to be clear, is ADCFB provides data back to the sites, but it also creates a resource for broader use in the research community. In CLARiTI, we have specific analyses that we're obviously going to do for the biomarker assay lab, but there is also the plan to do a wider range of biomarker assays as part of this study. And as you'll see on the next slide, if we try to have a participant do both ADCFB or use the ADCFB for the CLARiTI, we're actually reducing the opportunities to do research that we want to do as part of CLARiTI. Let me go to the next slide. So we recognize, and we've certainly heard, that despite what I've just said, the advantage of having a participant do both, there are going to be instances where you won't be able to have a blood draw for both ADCFB and CLARiTI. So NAC has come up with a good way that we can document that, and I'll explain how that will work. Sites, if they've seen a participant as part of ADCFB in the previous 12 months, so longer than that, this is not possible, but if it's within the past 12 months and there's no CLARiTI collection that's possible, then what we need to do is document that there is that prior ADCFB collection. And we need to be able to be sure that we can document that that's occurred. And we now have a form that's been developed by NAC via the CLARiTI case report form process. What you'll need to do is document that that previous blood draw was collected. You'll need to provide the date and to document that it was in the past 12 months. And that will be the form that will then alert us at NCRAD to identify that that sample was collected, and we will take a portion of the sample that was collected as part of ADCFB, the plasma, and move that to the CLARiTI study. I just want to stress that it's only going to be a smaller volume. It'll limit the analyses we'll be able to do in CLARiTI, and it will also reduce the ability to share valuable plasma with other researchers. So this is not our ideal solution, but we recognize there are instances where this is going to be needed. Let's go to the next slide. All right, it's up to you now, Jeff. Thanks so much. And I'll take questions at the end.
Yeah, thanks, Tatiana. You can go on to the next slide. So we're going to talk now about what we're going to do with the plasma samples once they've arrived at NCRAD. And so most of you are probably familiar with the ATN framework that first came out around 2018. And that's really to using imaging and CSF, categorizing people on their amyloid and tau status and neurodegeneration status. We're extending that into plasma, and this is the framework we're currently using. If you go on to the next slide, the way we're doing that within the biomarker lab is we're using amyloid beta peptide ratio, the A beta 4240 for the amyloid, tau or neurofibrillary tangles, we're using phosphorylated tau measures, and then neurodegeneration, we tie that to neurofilament light. And then we also are currently measuring glial fibrillary acid protein, or GFAP, and that's about astrocytosis or astrocyte activity, or a marker of astrocytes in general. A lot of work is still going into what that exactly is associated with. But you can see in the graphic that these four markers, the ATN and GFAP, they're measurable in both CSF and blood. So we're focused on the plasma samples that are coming for CLARiTI. And this is the same set of assays that we're using in the ADCFB, as Tatiana mentioned. So there will be a commonality in these assays. As Sterling pointed out at the beginning, one of the big goals for CLARiTI is to really help enable additional characterization of mixed etiology dementias. And for that, we need reliable biomarkers for some commonly occurring or co-occurring pathologies, such as Lewy bodies, TDP-43, or even pathology associated with vascular dementia. And so these are all research areas where we don't currently have assays or measures in the biomarker lab that we offer routinely. So we can go to the next slide. I think, oh, next slide, yep. So this really gets us into kind of the idea on the draft update to this framework that's currently under review and out for comments. I think you can still give comments. And this is really looking across staging, across additional pathologies to try and provide a better framework, a more extensive framework beyond the ATNN. And really, this is both the staging aspect, so in the core 1 and core 2, as well as in biomarkers of non-AD co-pathologies, which you can see in the bottom. So the vascular, synuclein or Lewy bodies, and then with imaging, you can look at anatomical locations of white matter hyperintensities. So if you go to the next slide, the biomarker lab, I'm going to kind of lead into this. The biomarker lab focuses on robust and reliable measures. And to do that, we can't really be switching assays all the time. We can't take on new assays that aren't really haven't demonstrated consistent clinical utility. And part of the reason for that is the way we do our quality measures and our bridging. So that's the bridging and control measures. We have pooled plasma samples that we implement as control measures, and we do qualify our assays so we understand the performance that we can expect out of those assays. And then we have this lot-to-lot bridging and even instrument-to-instrument bridging that allows us to run samples across time and not have to absorb batch effects. And so it allows us to kind of do this ADCFB, or in this case, CLARiTI, where we're going to run samples, quarterly or semi-annually and give them back to the sites and to be able to have that data used for analyses. And it's the only way we can kind of have that continuity is to have a really well-controlled assay. And so that's one of the reasons we can't take on new assays really rapidly, and why we wait for some reliable assays in terms of their clinical utility. So go to the next slide. I wanted to give you a snapshot of what this means. So I mentioned our reference pool. So we collect plasma from participants here at IU School of Medicine, and we create reference pools for each of our assays. And we have 3 pools that we'll use in each assay. And this is just kind of showing you that for GFAP. And we have these 3 pools that we include in every batch and sometimes on every plate within that batch, so that we can kind of keep track over time. We can look for variations, we can look for drift in our assays, and we can look for bias that might show up because of lot-to-lot changes. And now this is showing this over I think almost 200 plates and I think it's well over 6 months. But it's a lot of samples and it's a lot of time, and there's lot switches that go on within that time that we have to bridge. And so when we do that, we adjust the calibration so that we don't have bias due to manufacturing lots. And again, this is something that's necessary if we're going to have longitudinal measures. So if you go to the next slide. These are the assays that we currently have operational. So the ones that say qualification status complete. The ones in the green boxes are the ones that we're implementing initially within the CLARiTI program. But we do change over time. When we do change either platforms or assays, we do perform bridging assays. We do comparison tests to make sure that we're going to be able to give you instructions on how to utilize that data in combined analyses. And so right now we're using the Quanterix Neurology 2-plex, the N2PB, and we use that to measure neurofilament light and GFAP. And then we're using the Quanterix provided ALSPath phospho-tau 217 assay. And then we measure the 4240 amyloid peptides. That ratio we measure on the Fuji-Radial LumiPulse platform. And we found these to be really well-performing assays. The 217 seems to be a little bit better than the 181s, and there's been several papers out. The LumiPulse 4240 does perform a little bit better according to the papers compared to the one that's included in the Neurology 4-plex. So I think we have the best set of performing assays that we're currently leveraging, but we do have quite a bit of data comparing these and bridging sets across these different platforms. And this is where we will start with CLARiTI. So if you go to the next slide, where are we going with CLARiTI? So we mentioned wanting to do more novel testing. There's two types of novel testing. One is on the novel biomarkers. So I mentioned that the field is rapidly evolving. The biomarker assay lab within NCRAD isn't as equipped to take on the cutting edge or the bleeding edge of those biomarker assays. But we are positioned to rapidly implement assays that demonstrate reliable clinical utility. And so for that reason, there's been several different assays coming out looking at soluble oligomeric forms of synuclein, or even amyloid beta or tau aggregates. And we're keeping our eye on that. And we will, once we get enough samples within CLARiTI, we'll probably be taking a look at one or more of those assays. And then there are new assays coming out, which are quite exciting, looking at cryptic peptides associated with TDP-43 pathology. In both cases, the synuclein and the TDP-43 seem to work a little bit better in cerebrospinal fluid than plasma, but we're keeping an eye on this field, and hopefully it's going to be developing over the coming couple of years so that we'll be well positioned to pick these up very rapidly. And then there are some new papers out just recently on evaluating different preparations of using the plasma to purify extracellular vesicles, and then looking at the packaged materials within those vesicles. And that's another area that we're currently watching. All three of these types of areas, or these pathology measures, we're just watching, and we are going to select assays to pilot as we start to collect samples within CLARiTI. The other thing is the graphic on the right is this idea of remote collection devices. There's been a couple of papers looking at this. Well, one from Nick Ashton, where he was looking at dried blood spot analysis across these same assays and looking at the performance. And it shows some promise. And so in order to make these biomarker screening tests, to be in these biomarker tests in general, to be enable accessibility of typically underrepresented groups, people in rural populations, people who live quite a distance away from some of these centers. We do want to look at this technique, but it won't replace what we're asking for in terms of standard venipuncture and shipping of plasma within CLARiTI. But CLARiTI is positioned to do these kinds of studies and enable this within the protocol. It's actually within the protocol already. And we'll be building up to implementing this as the technology emerges. So I think we go to the next slide. And this is, this is the CLARiTI contact for NCRAD. For all things, you'll probably use Micah Gosnell mostly for the stuff Tatiana talked about in the kits and the tubes and the shipping materials. If you have questions about the biomarker assay lab, the results or things like that, you can contact myself or Dr. Kristen Ross, the director of the biomarker lab. And I think that's our last slide. So I think we move on to Dirk to talk about neuropathology.
Thanks, Tatiana and Jeff. I'd love to share my screen if you could stop sharing that screen. Thank you very much. So, hello everyone. This is let's see, can we do this? So this is my opportunity to sort of introduce everyone to the Neuropath core. I'm really excited that Sterling and Beth and everyone had the foresight to try to include Neuropath in this really massive endeavor. That isn't always the case. And so we are looking far into the future, I think, with this core, but starting now, and that's particularly exciting. Why Neuropath? I think for a couple of reasons. One, as Sterling mentioned, this is really not just about Alzheimer's disease. This is about the polypathology that we see and how to interpret the biomarkers that are coming from people with these heterogeneous pathologies. And so Neuropath is still the gold standard for really diagnosing what is happening in these folks. It's a point in time, and we have to extrapolate, and we have all kinds of limitations that way. And so having these new techniques that Jeff just mentioned is really exciting, and I think ultimately is what we'll be relying on. But we need Neuropath to help us understand what we're seeing as we go, and as these new markers are developed and tested in this banked material. I think the neuropathology of these folks is going to be really important. So you can imagine that we haven't enrolled anybody yet, and so the idea that we're going to have Neuropath is really a sort of a future thought. But we have a plan, and we have, I think, something in place that we've proposed that I think will work. This is sort of the general overview of what Neuropath is going to do in CLARiTI. And so sorry about that. Let me try to put up a pointer here. So the ADRC NP cores will be the boots on the ground for any CLARiTI participant who dies, and they will presumably, if they have consent, follow their normal protocols at each of the ADRC sites, which involves at a baseline at least the NIAA guidelines for assessing neuropathology, and I'll get to that in a second. The idea, just to summarize with this slide, is that the ADRC Neuropath cores will be providing Neuropath data using the NIA guidelines and the UDS to NAC, and hence to the rest of us, and that they will be providing either slides or blocks or wet tissue to the Neuropath core, the CLARiTI Neuropath core at UW, from regions that are being dissected anyway. We're not asking, I don't think, for a lot extra, where we will develop basically a core resource for CLARiTI, a reference data set that's going to have a lot of the same stains, although not completely, but where we can digitally scan and share all of this data from these slides from each of the sites back to the sites, and then through the network through NAC, and then leverage some of the work that we're doing here in conjunction with what I hope is a really robust consensus process across the Neuropath cores throughout the network, to try to develop quantitative approaches and interpret the results that we're having, while along the way, I think, testing as a group different aspects of, inter-rater reliability, the variability that happens not just from staining across the network versus a centralized site, but how that influences the quantitation that we can do. And I'm hoping that we will work together as a group of Neuropath core leaders and neuropathologists to sort of help develop this into something that it turns out to be really robust. And that'll happen formally through a consensus process, but hopefully more informally along the way as we interact. So just to sort of summarize the goals, like I said, the goal is really to develop this reference set, to have that, I guess I can still say the ground truth of what's happening in a person's brain once they die, to be able to interpret what we've seen through CLARiTI over these longitudinal biomarker measurements, whether it's imaging or biofluids or cognitive data. It's going to be really important to have that neuropathology reference set. And we'll get there through a variety. I'm not going to read the slide to everybody, but we have to sort of build a Neuropath core network that is essentially already in place through the Alzheimer's centers, but we need to work together a little bit more than we do to sort of build this resource out. And I'm hoping that we'll have really great engagement through this collaborative network that we're going to develop in these pipelines that we'll have to sort of build so that we can develop this resource. And of course, there will be CLARiTI participants who are donating their brains 10 years from now. And so this is the first step, right? We want to build this, but this isn't just for CLARiTI. This is something that we hope will be durable with CLARiTI or even beyond CLARiTI, so that we can ultimately have a really good sense of what's happened in most, if not all, of the CLARiTI participants from a neuropathological perspective. I want to introduce Dr. Erica Malif. She is a scientist in our group who is really well versed in quantitative neuropathology. She leads our quantitative neuropathology team, which is pretty large actually, and has taken on the role of project manager for the CLARiTI Neuropath course. So you'll get to know Erica a lot more if you're involved in a Neuropath core for sure. We're just getting this off the ground, obviously, so stay tuned. So how does it work? So like I said, a CLARiTI participant dies, and we aren't requiring participants in CLARiTI to have a neuropathology, a brain donation consent for a lot of really good reasons, but most of them hopefully will. That's a goal of our Alzheimer's centers. And then the ADRC will perform their usual autopsy and neuropathological evaluation, and all of this is sort of based in the NIAA guidelines that were published 12 years ago now. We all do something a little bit differently. We can use different stains, we can do different approaches, and a lot of us do a lot more than the NIAA guidelines require, but we're trying to build the CLARiTI Neuropath core based on the NIAA criteria and the UDS that we're required to provide at a minimum for all of our brain donors in the Alzheimer's Center network. And so I think most people know that the neuropathology is based on the distribution of beta amyloid plaques, that's the Thal phase, on the distribution of neurofibrillary degeneration, that's basically neurofibrillary tangles, and that's a tau measurement, that's a Brock stage. And then on the density of neuritic plaques, the cortical density of neuritic plaques, that's the ABC score, that's Thal, Brock, and CLARiTI. And so all this data is going to be coming automatically from CLARiTI participants who donate their brains. And so we don't necessarily need to repeat that. The thing is, it's going to be coming from 37 or 50 or however many neuropathologists that are throughout the network. It's going to be coming with a variety of different stains. And we have an opportunity through the CLARiTI Neuropath core to extend the work that was done after the NIA guidelines were developed by a core group of amazing neuropathologists. I was not one of them, but they really started to ask questions about, variability across sites and with digital pathology. I think we can extend that though, with a really robust resource that connects back to incredibly well-characterized, longitudinally characterized participants. So this is the minimum sampling protocol that is required in the NIA guidelines, right? We have basically 13 brain regions. They're all just sort of depicted here on one side of the brain, but this is the minimum sampling that we all do. A lot of the Neuropath course frees one whole hemisphere, and so we don't get these from both sides. A lot of us take it from both sides and have different ways to process the brains. But at a minimum, we would want one hemisphere samples from the core regions that are used for the NIA workup, and that would be this. And then, as you all hopefully you may know, the NIA guidelines have this very complicated algorithm for how we stain the slides. At our site, we just do it all at once. We don't try to do the if this then that. Some other sites will take a different approach, and the antibodies are very flexible. There's a lot of different ways we can stain for neurofibrillary tangles, a lot of different phosphorylated tau antibodies, different ways to do A beta and alpha-synuclein, of course, TDP is part of this as well, which wasn't a part of this when we first started. And so that's the NIA guidelines, what we're proposing, and this is complicated, so I'll try to simplify it. For the Neuropath core, for each site, we've simplified it down to 12 brain regions that should automatically be sampled from every site that are hopefully easily either double sampled as far as a wet tissue sample or a paraffin block, or we can get unstained slides from each of these regions from each of the centers for each of the CLARiTI participants after they've died and they've been their tissues have been processed and worked up. And we're asking for 8 slides per region. That's a total of 96 unstained slides per donor. We do have funds in the Neuropath core to support this at each site, so we're not asking for this for free. And what we propose is that at the Neuropath core for CLARiTI, we're going to perform H&E LFBs on everybody, on every block. We have a pretty robust set of A-beta stains. p-tau, we use AT8 here for the Neuropath core. We have alpha-synuclein, we use LB509, but there are other, certainly other options. And then we use the 1D3 clone for phospho-TDP. And so this is just summarizing the number of stains that we would do routinely for every CLARiTI participant in the Neuropath core. Why do we do that? Well, basically, this allows us to perform a very comprehensive NIAA workup for each of these donors using harmonized, centralized staining. If that's something that we decide we want to do as a group. I don't know that we want to redo the NIAA staining or guidelines for everybody. What we do want to do, though, is we want to digitize this so that this centralized, harmonized resource is available as a CLARiTI resource for the CLARiTI cohort. And so that's where I think I'm really excited about what the Neuropath core for CLARiTI can do. We have a reasonably robust system set up here. Again, Erica leads these efforts at our site. We basically are using Apereo as our scanner. We use Halo as our image analysis application. We found that Halo works really good in sort of large, high throughput situations, but gives us a lot of flexibility, especially the AI classifier options, which really have enabled us to hone in on techniques that are good for evaluating or classifying, annotating cortical layers, for optimizing for the different stains and the different aspects of the stain, whether it's, typical percent area or, frequency or number of events, or more nuanced things like activated microglia. We propose again to do these typical stains across the typical NIAA approach, but what we are working on through some of our other efforts is a larger set of stains, these 7. So we're able to assess neurons, astrocytes, and microglia, which I think again gets back to a lot of the work that Jeff is working on and that is building through these biofluids in a manner that gives us a robust way to with 30 plus quantitative measures using these stains. And what we've done, just as an example of the power of this is from our work in the Seattle Alzheimer's Disease Brain Cell Atlas that we work on with Allen Brain Institute, led by Ed Lean. What we've done is we've taken all of these metrics from those 7 stains that I mentioned earlier, and we've used this across the 84 donors. Each of these columns is a donor, to sort of use PCA to rank order these donors based on their aggregate neuropathology, the quantitative neuropathology. And so what you can see here is this is the order of the donors. These are, of course, related because these are the measures that we're using for the quantitative analysis. And so we can see good correlation with what we would expect to see. But what is important is for the cognitive outcome, the CASI, which is basically a fancy MMSE. For what we measure is AD neuropathologic change. And all sort of rank orders these folks in a way that gives us much more power, much more effect sizes to detect differences. So this is the single cell RNA-seq data, the transcriptomic data from single cells. These are the different cell types that are based on the Brain Initiative work that the Allen Institute is doing, and using cognitive status to look for effect size, or ADNC as we would calculate from NIAA, gives us a pretty robust assessment of certain cell types that are decreased or increased in the case of glia. But what you get when you get this continuous pseudoprogression score developed from this quantitative set of variables is just a larger effect size. So it gives us more power to detect more subtle differences across these different cell types. We can extrapolate this to biofluid data. We can extract this to imaging data from these donors and to try to understand how the different volumes across different regions of the brain or different, PET, densities are related to these different quantitative pathologies. We can also do this across brain regions. This is dorsolateral prefrontal cortex here. This is middle temporal gyrus, to see how this varies across the brain. And that's a really important contributor, I think, to what we're going to be seeing with ATN, is how much heterogeneity there are across regions and how much burden there is across the brain. And so the other thing that the continuous pseudoprogression score does with this quantitative approach is it gives us the ability to measure pseudo time, which has always been a real challenge for me as a neuropathologist. We're looking at a point in time, but we can interpret based on how much pathology a person has, where they are as far as the neurodegenerative condition for the different cell types. And what we found with CAD is that we're seeing early losses of inhibitory neurons that we wouldn't necessarily expect given where we see neurofibrillary degeneration in tangles. And so this is a really interesting way to start to probe what's happening in these people over time across multiple brain regions, and how does that correlate, even though it's a point in time, with what's been happening over the last 3, 5, 8, 10 years, because we have all this biomarker data. And so we propose to build a data set from the slide scanning, from certain brain regions, from the CLARiTI reference set, and to provide that, of course, to NAC and to the sites based on the cases. And then to just wrap up, once we've got that all rolling, I hope to have meetings with as many of the Neuropath core leaders and stakeholders who are interested, probably quarterly, to update our progress, to coordinate what we're doing, and ultimately, through consensus, but certainly on an ongoing basis, start to think about how we can really use this information to advance neuropathology of AD and ADRD, but also to really inform what we're seeing from the incredible characterization that we're having with CLARiTI. And so I think some of the options are, of course, going to be understanding differences between centralized and diversified neuropathological assessments. That's kind of low-hanging fruit. How can we incorporate quantitative pathology or digital pathology across the network beyond what I've just talked about, to really help us understand and measure and characterize the brains that we have, and again, understand what's happened before these people have died. And, are there better ways that we can be assessing neuropathology in these folks that will help us distinguish more clearly what was happening in these donors? And that's especially important when it comes to polypathology, right? We really need to have a good handle of who's got what when they're coming into the clinic, using neuropathology as our guide so that we can better tailor our treatments. And I think that's ultimately what this is all about. So I think there's a lot of potential benefits. I think I've gone over all of them essentially, but we're hoping to build a really great resource and to have input from everyone to help us figure out how best to use this resource and learn from it. So with that, I will stop.
Thank you very much to our presenters. All right, we will open it up for Q&A. We've covered everything. No one has any questions.
There is one in the Q&A box.
Oh, it's not showing up on my end.
Oh, here, let me adjust that.
Thanks, Hannah.
I can read it though. So Carol says, I'm surprised to see plasma frozen at 1.5 milliliter aliquots. How will freeze-thaw cycles be minimized given microliters of samples are generally needed for experiments?
I'm glad to. Jeff, do you want to answer? You want me to do it first?
You can go first.
Okay, so what we do is, historically, this is what we do at NCRAD. We go with 1.5 mils because it's sort of a balance between site burden and exactly what you're describing, which is freeze thaw. So what we'll do here is we will sub-aliquot the plasma. We'll only do them 1 tube at a time because we recognize that there are changing demands on how much plasma we need for biomarker assays or for other types of assays. So currently we will just sub-aliquot the first 1.5 mil tube. That will give us the volume that we'll need for the biomarker assays that we'll do here in the ball. and the remaining volume will go into 200 microliter aliquots. We leave the other 1.5 mils alone precisely for what you're describing. So as the demands for plasma potentially reduces, then we'll be able to go into different types of aliquot sizes. So it gives us flexibility without having the sites create large number of aliquots, which we've historically seen as a burden, and sites struggle to be able to do that accurately. I don't know if you want to add anything, Jeff.
Yeah, I'll just point out, like, I think that uncertainty is the key thing, and starting in the 1.5 volumes is a really great approach, in my opinion. But it's not only freeze thaw, it's also time in the freezer to consider. And some people, when they do low volume aliquots, they have large headspace. And within the freezer, you'll get sublimation and you get concentrations of biomarkers. So, depending on how long things sit in the freezer. And so what you might think is like a longitudinal study ends up being just a time and freezer study showing you increases over time. And so there's a lot of confounding effects that you have to worry about, not just freeze thaws. And this approach really kind of balances that. So it allows you to go into a lower volume, lower headspace kinds of aliquots when needed, or distribute just a few microliters to somebody who needs it at the time of a single freeze thaw. So I think it's a good balance. And since a lot of the things, the new ones with synuclein, TDP, and vascular markers, we're not sure yet the volumes that are going to be required, if they're going to be like the 1 microliter or they're going to be the 2 mil variety. We just don't know. So, I think this is a nice starting point.
Great, thank you. I see somebody has their hand up. If you'd like to unmute, it's YF Dotson. If you'd like to unmute and ask your question.
Yeah, I got on late. I'm sorry, I was in the clinic. Dr. Foroud, you started off talking about the blood draw, and that'll be my end of the visit. And I got on late. How many extra tubes do we need to draw? This is Yolanda from Clinical Core.
No problem. So for the CLARiTI blood draw, it's a 30 mil blood draw. So you would do 3 10 mil purple top tubes.
To add on to what we're already drawing, so those will be in addition to?
That is correct.
Okay.
I also did discuss the situation, which isn't our preferred, which is coordinating an ADCFB collection with a CLARiTI collection.
And we could also add that you can do the CLARiTI blood draw at the time of the imaging visit, if that's more convenient for the participant and for you.
Absolutely.
Do they need to be fasting?
You'll record if they're fasting or not. We recognize, depending on the time of that imaging, it may not be possible.
Well, we ask them to fast when they come in for us. So if they come in for their core visit and they agree and everything, they're already fasting. But I didn't know, like, if we're doing it for imaging, do they need to be fasting?
No.
It's not required.
Okay.
I just want to be clear that we do ask you to record whether or not they are, though. Just to be clear, we're going to want to know if they were or not.
And are we going to have an extra form to fill out for that? Will there be an extra blood draw form? And it still comes to you at 835?
There will be a separate case report form for CLARiTI blood collection. Yeah, thank you.
And you'll send me where to send it to and stuff?
Yep, we'll have training on that.
Okay.
Thank you. All right, if there are no other questions, Phoebe, last slide, please. Oh, thank you. I see that our next webinar is going to be led by Dr. Gil Rabinovich, leading our visual reads course. So that'll be on August 28th. So we're taking the month off of July for AAIC. So hopefully we see many of you there. And then just some reminders how to stay in touch with us. You can always email us, clarity@medicine.wisc.edu. I will mention that for those sites that have Hanja as their project coordinator, he's out through the rest of the month. He's got a sweet little baby at home. So if you do email Hanja, please also email clarity@medicine, and then somebody from our admin team will help you with your request. You can email the inclusion team, and then please refer to our website as always. So we'll end there. Thank you everybody for joining us. Thanks so much to our presenters. Take care, everyone.
Thank you, everybody.
Yeah, thank you.